Yeah, so I'm Mark. I'm a postdoc in the mathematics group, mathematics department here. Like Ron said, I'll be talking about stability-based sequence optimization via tensor networks and dynamic programming, as we'll see. So in general, in this talk, I'll be showing how we've given a new approach to nucleic acid design, using tensor methods. The basics of the particular problem that we're treating are the codon optimization problem that we're focused on is that we want the best mRNA sequence which encodes a given protein, which is an input to this problem. To clarify the terms here, we're designing an mRNA, so that's just an RNA, which is a sequence of bases A, C, G, and U. And we're going to design this mRNA such that it codes for a protein. The protein is just a pre-specified long chain of amino acids in this context. And the way that this coding in biology works is very simple. Every sequence or word of 3 consecutive RNA bases in that mRNA code for an individual amino acid. On the other hand, multiple codons can code for the same amino acid, as is shown in this codon table for the natural amino acids shown on the top right. And so this gives us a lot of flexibility such that we can choose many different potential RNA sequences, all of which code for the same protein. On the other hand, some mRNA sequences are thought to translate better than others, and one of the particular metrics that people use is a concept of stability. So this will be the one that we focus on. And then while this problem has clear applications in biology, it's actually a very widespread problem and applications here at LBL across DOE. So it's used for metabolic engineering and biofuel production, used in chemical synthesis, and this is all besides the obvious relevance to use in vaccines and cancer treatment. So as I mentioned, we're going to focus on nucleic acid stability as a metric of the mRNAs that we design. We can analyze, for a specified RNA sequence, we can analyze its stability using tensor networks. The reason that we want to look at stability in general is that more stable mRNA sequences are correlated to having stronger base pairing. And if there's more base pairing, then it's thought that this mRNA will be less prone to interference from other molecules within the cell, so then it will be translated better and make more protein at the end. So in particular, the way that we model the stability of a given RNA sequence is by considering all of the different secondary structures that it can exhibit. The secondary structure can just be represented as a list of base pairs. So basically, we have an mRNA sequence, as is shown on the top left here, which you can view as a sequence of codons. Each codon is a sequence of 3 bases. Then all sorts of different base pairs can form between different bases along that sequence. If we lay the sequence out along a line, those base pairs can be visualized as the arcs on the top right. We can also contort that diagram into the one on the bottom left, showing a more physical description of how this molecule might actually look, at least in a simplified 2D way. And then for this individual secondary structure, we can model its stability in a very natural and appealing way using tensor networks. So here, the diagram on the bottom right is a tensor network. Contracting that tensor network gives the free energy of the structure on the left. So we've demonstrated in past work that tensor networks can accurately model these free energies, all for a given RNA sequence. But we want to move to this different application in which we design RNA sequences. So here the RNA sequences are really variables. And at the end of the day, we want to choose a single RNA sequence using our algorithms. For the first time, we did this using a concept of tensor trains, which we've heard about in other application areas today. A tensor train is a simple probability distribution over a sequence of variables, each of which conditionally depend on variables right around it. And what we've realized is that we can use tensor trains very naturally to enforce the codon constraints of our original problem, ensuring that any of the RNAs that we design code for that protein that we're interested in. Moreover, there are a large variety of other sequence-based objectives that can also be modeled using tensor trains. And a variety of them can be found in the literature, and we can make tensor trains which model basically all of these in different ways, some of which are shown on the bottom of this slide. But if we have a tensor train which is showing, just modeling these sequence-based objectives and the hard constraints of codons, we still need to combine this with concept of stability. And as I mentioned, we measure stability by looking at the contractions of different tensor diagrams for the different secondary structures that a sequence can exhibit. So for a fixed RNA sequence, we've previously demonstrated that each of these diagrams for a secondary structure can be contracted in order n time. While furthermore, we can sum over all secondary structures, which is an exponential number of structures in order n cubed time. But what we do in this project is that we can combine tensor trains with these secondary structure diagrams. So this is shown on the right, that have a tensor train shown in purple and a secondary structure diagram shown in blue and green. If we contract these together, we can lay out the tensor train along the backbone of the RNA strand, and then combine all of these degrees of freedom by recontracting them, such that we get a diagram like the ones on the right. And these naturally exhibit the same topology that we started with in terms of the original secondary structure diagrams. What all of this means is that we can use almost precisely the same algorithms for a fixed RNA sequence as we can for a variable RNA sequence. And when we plug in a variable RNA sequence, this is how we can do design. And so this gives us very naturally algorithms that are very similar to past ones that I've developed, but only incurring a roughly 4x slowdown for the problem of design compared to analysis. Moreover, this approach is very principled and allows us to compute a variety of exact statistics. So if we want, in our approach, we design, in the end, a sampler such that we can sample an RNA sequence out with that'll solve our optimization problem to high probability. With our approach, we can compute the marginal codon probabilities exactly, just the probability of a single codon being sampled at a given position. We can also compute base pair probabilities analytically. So this plot on the bottom right is showing us for our design sequences, where are the regions that are likely to be unpaired or base paired. And the unpaired domains are more likely to be attacked by RNases and other molecules within the cell. And then we can finally compute a minimum free energy sequence, replacing our sampler with an argmin operation, such that we get a single sequence that solves a minimization problem. So all of this indicates that in our approach, is based on a direct and IID, or independent and identically distributed closed form sampler. And so not only do we just get one sequence, we get a whole ensemble of favorable sequences. So using these metrics of stability on the top and base pairing probability at the bottom, we show that the ensembles of sequences that we design are superior to those of competing approaches. In terms of performance, the tensor-based design is also fast and GPU compatible. So the dominant CPU expense or dominant expense of these algorithms can be phrased in terms of triangular matrix multiplications. And so this is simply computable via standard Labs routines and is easily parallelizable. And so GPU acceleration, once plugged in for these operations, can offer over 100-fold speedups. So all of the optimizations, even for sequences of over 10,000 nucleotides, can be done in less than a minute. So with that, I hope I've illustrated how this kind of discrete math is powering new methods for sequence design. Our future extensions that we intend are for structure-based design, where the designed RNA has a desired structure known to experimentalists, and then also experimental collaboration to try to deploy our algorithms. With that, I will thank you. My primary collaborators are Christina Wang, who is an intern here, and Michael Lindsay down on campus.